polling, we'll go straight to Tim Yap's talk, Targeting DNA Damage Response and HRD in Breast Cancer.
Good afternoon, and thank you all for being here. I'd like to thank the organizing committee for giving me this opportunity to discuss a topic that's very close to my heart, and that is targeting the DNA damage response and HRD. These are my disclosures. So when I think about targeting the DNA damage response, or DDR, I like to think about them in different buckets. For example, targets along the DDR pathway, such as PARP, ATR, and ATM, or those targets along the DDR cell cycle, such as CHEK1, MYT1, or WEE1. And what is important is that we currently have multiple different agents targeting the DDR in the clinic, as listed here. We have seen success with PARP inhibitor monotherapy in breast cancer, including olaparib and talazoparib in metastatic germline BRCA1 and 2 mutated breast cancer, as shown by data from the Olympiad and BRCA studies. Most recently, we've seen the data from the Olympia study of olaparib and adjuvant germline BRCA1 and 2 mutated breast cancer. But we need to build on this success, primarily because not all patients with BRCA1 and 2 mutated cancers will respond. Drug resistance is nearly inevitable even if you respond. And there is certainly great potential to deepen the responses, potential to deepen the actual durability of response, and also potential to widen the application of DDR inhibitors. And the good news is that the DDR landscape, certainly therapeutically, has been rapidly expanding beyond PARP inhibitors. We now have discovery of novel precision targets that has been enabled by cancer genome sequencing and modern CRISPR technologies. And these strategies have essentially allowed us to undertake biomarker-driven patient selection to guide the clinical development of many of these different agents. And these include beyond PARP inhibitors, ATR inhibitors, WEE1, ATM, DNA PK, CHEK1, and RED51. We also have the new kids on the block, the new kids in the clinic, such as PK-MYT1 inhibitors and pol theta inhibitors. And a point I really want to get across is that the DDR synthetic lethal landscape is much bigger than just BRCA-mutated cancers and PARP inhibitors. We're certainly just at the tip of the iceberg. For today's talk, I'll just be focusing on ATR inhibitors, PARP inhibitor combinations, and the new kids on the block, PK-MYT1 and pol theta. So here's a quick overview of my talk today, targeting ATR, rational PARP inhibitor combinations, and then PK-MYT1 and pol theta inhibitors. So what is ATR? Well, ATR is an apical signaling kinase found along the DDR pathway. It is activated by stalled replication forks or replication stress. And ATR has a key role in the DNA replication stress response pathway by facilitating the recovery from stalled replication forks and preventing premature mitosis. So here's the ATR inhibitor therapeutic landscape. You can see it's extremely busy and also a rapidly growing space. There are multiple agents. First in class was the IV compound pazosertib from EMD Serono, which is currently in phase 3 trial testing in combination with chemotherapy. We also have other oral agents, ceralasertib from AstraZeneca, lumasertib from Bayer, and RP-3500 from Repair. We also have newer agents such as the Artios compound, which is currently in phase 1 trial testing, as well as the EMD Serono compound M1774. The Atrin and Impact compounds have recently entered or are about to enter the clinic. So as we think about potential registration strategies for ATR inhibitors, there's certainly monotherapy or combination approaches. For example, with monotherapy approaches, we should think about the biomarker, for example, by moving the DDR label beyond BRCA-mutated breast cancer and focusing, for example, on ATM loss-of-function tumors. We can also think about targeting PARP inhibitor-resistant cancers, such as olaparib or talazoparib-treated BRCA-mutated breast cancer. And there's certainly strong and robust preclinical rationale that support this. In terms of combination approaches, There are certainly chemotherapy or ADC combinations that look promising, for example, gemcitabine plus ATR inhibitors. There are also DDR combinations, for example, PARP inhibitors plus ATR inhibitors in a PARP after PARP resistant setting. There are also IO combinations with PD-1 or PD-L1 inhibitors, and then a chemical BRCA combination approach using targeted agents to induce BRCA in HR proficient tumors to try and synergize them with ATR inhibitors. Let's talk about monotherapy first. I've drawn here on the left-hand side a very normal-looking ATM proficient cell. You'll see the intimate interaction between the ATM and ATR pathways. ATR, as mentioned, is a key regulator in response to DNA damage and replication stress that promotes DNA damage repair and survival. On the right-hand side, in contrast, you'll see an abnormal-looking ATM deficient cell, and you'll see a dependence on the ATR pathway. And therefore, the good, strong rationale here is that ATR inhibition in cancer cells with certain DDR defects, such as ATM alteration as shown in this figure, may actually induce rapid cell death and therefore lead to synthetic lethality. And certainly we have seen early signals of response in ATR inhibitor studies, especially durable responses in ATM lost cancers as shown here in the figure. I show here a spider plot on the left-hand side with the green curve showing partial responses, and on the right-hand side, the responses observed on this trial. Most of these patients had ATM loss of function. You'll see that an HR-positive, HER2-negative breast cancer patient who achieved a partial response, minus 54%, for 356 days. And you'll see here that the durability of the responses observed on the study were pretty impressive in those patients who responded. The key question is whether ATR inhibition can actually overcome PARP inhibitor resistance. As mentioned, there are strong preclinical rationale. For example, I show here two papers, one from Yves Pomier's group showing that resistance to PARP inhibitors by SLFN11 inactivation can actually be overcome by ATR inhibition. On the right-hand side, Li Zhu's group showed that ATR inhibitors can actually disrupt the rewired HR and FOLC protection pathways in PARP inhibitor-resistant BRCA-deficient cancer cells, therefore supporting the use of ATR inhibitors in PARP inhibitor-resistant setting. And we are now seeing early signals of these activity. I show here data from the phase 1 TREASURE trial of the ATR inhibitor RP3500 from Repair. You'll see that we saw responses in the post-PARP inhibitor setting in 4 BRCA-mutated patients and RAD51C mutated cancer patient, and also in a response in a BRCA1 mutated ER-positive breast cancer patient, amongst other tumor types as well. Moving on to rational PARP inhibitor combinations, I'd like to divide these up into 4 different buckets. We have chemotherapy or ADC combinations, DDR combinations, immunotherapy combinations, and also molecularly targeted agents that induce chemical BRCA. Starting off with combining PARP inhibitors with ADCs, preclinically, PARP inhibitors and DNA topoisomerase 1 or TOP1 inhibitors have highly synergistic anti-tumor effects. We've seen this from robust preclinical data. However, the clinical development has really been hampered by overlapping toxicities, and therefore it was hypothesized that tumor-targeted delivery of a TOP1 inhibitor may actually reduce the systemic toxicity of the combination. As you know, sacituzumab govitecan is a TROP2 ADC. This is an anti-TROP2 antibody that's coupled to an active metabolite of irinotecan SN38 via a proprietary hydrolyzable linker. So we undertook a phase 1b trial in advanced TNBC patients and other tumor types, as well as patients harboring HRR alterations. Prior PARP inhibitors was permissive, and we started off at a dose of rucaparib 300 mg BID with sacituzumab 6 mg per kilogram IV on days 1 and 8. Here are some results that were presented at ESMO last year. Out of 6 patients, 3 patients achieved a RECIST partial response. All 3 patients received prior PARP inhibitors. You'll see that a TNBC patient who actually did not have any detected deleterious HRR gene alterations. And I show on the right-hand side a scan showing that partial response. Despite these promising results, you'll see here that the treatment-emergent AEs really included a high percentage of patients with grade 3 or worse, neutropenia, and thrombocytopenia overall. Moving on to DDR combinations, there's certainly strong mechanistic rationale to combine PARP and ATR inhibitors, and that's because PARP inhibitors cause increased DNA adducts which stall replication forks, and as mentioned, ATR is required for the repair of such stalled replication forks and replication stress, and therefore in conjunction with, for example, ATM deficiency, the combination of PARP and ATR inhibition will lead to increased DNA damage and cell death. And certainly right now, multiple PARP and ATR inhibitor combinations are ongoing. Moving on now to molecularly targeted agents that induce BRCAness. Well, the whole concept here is that you can actually enhance the sensitivity to PARP inhibitors by inducing an HRD phenotype in HR-proficient tumors with molecularly targeted agents. As shown here in the bottom figure, if you take an HR-proficient tumor, these cancers are driven by signaling pathways that promote HR and allow these tumors to evade apoptosis. And therefore, it would make sense that if we can actually intervene with potent and selective targeted therapies to induce an HRD, you'll then sensitize these HR-proficient tumors to PARP inhibitors and other DDR agents, as shown there. An example of such a targeted agent may be a BET inhibitor, which has been shown to be potentially active in tumors in combination with talazoparib that don't usually respond to single-agent PARP inhibitors. And this is important because there has been limited activity, as you know, of PARP inhibitor monotherapy in metastatic TNBC tumors that don't harbor BRCA or PALB2 mutations. And in TNBC, only approximately 20% of patient tumors have BRCA or PALB2 tumors. Importantly, the BET inhibitor ZEN3694 has been shown to reduce mRNA levels of several DNA repair genes as a potential mechanism of sensitization to PARP inhibitors by inducing the so-called chemical BRCAness. And BET inhibition sensitizes tumors with functional BRCA1 and 2 and PALB2, therefore expanding the potential use of PARP inhibitors in TNBC. And some of these data were presented at this meeting last year. And thanks to Zenith, here are some data from the Phase 1b/2 trial of ZEN3694 plus talazoparib in combination, showing clinical activity in this waterfall plot in BRCA wild-type TNBC. You'll see an overall response rate in the overall population of 22%, but this goes up to 32% when they divide it up by two different subtypes. Subtype 1 leads to 32% response rate, but 0 overall responses in subtype 2. Zenith have not revealed this subtype just yet, so watch this space. In terms of combining PARP inhibitors with immunotherapy agents, for me, the key thing is that there are so many IO agents in combination right now, greater than 1,000 PD-1 and PD-L1 inhibitor combinations right now. It's almost like PD-1 inhibitors have become the chocolate of oncology because chocolate makes everything better, but we know that this is not true, and we really do need to stop the serendipitous development of combinations, and it should really be biology that's driving the development. And I do believe we have this with PARP plus IO inhibitor combinations. There are different hypotheses, of course. The initial one was that PARP inhibition would lead to S-phase DNA damage, increasing neoantigen expression, creating a more antigenic immune microenvironment. A second hypothesis, which I prefer, is that S-phase specific DNA damage leads to an accumulation of cytosolic DNA. This activates the CGAS sting innate immune response, stimulates type 1 interferon, CD8 T cell recruitment, and leads to synergy between these two classes of agents. There are also other hypotheses, for example, PARP inhibition inactivates GSK3 beta, leading to PD-L1 upregulation and in vivo synergy. So choose which hypothesis you like, but ultimately for me, I believe that combining PARP and PD-1 or PD-L1 inhibitors is certainly a rational anti-tumor strategy. And here are some data from the Mediola study led by Susan Domchek that was published in Lancet Oncology last year. These are data from the phase 2 study of olaparib and durvalumab in germline BRCA-mutated metastatic breast cancer. And you'll see there an overall response rate of 63%. What is important, though, is that when you consider the published data currently of the combinations of PARP plus checkpoint blockade, for example, with the Topatio study, where they combine niraparib plus pembrolizumab, and you compare these data of the combination with single-agent PARP inhibitor data from Olympiad and also UMBRECA, you'll see that the overall response rate is probably quite similar, and that the addition of immunotherapy did not obviously increase the overall response rate. And in terms of where benefit may be seen in improving patient outcomes, perhaps this will be seen with increase and improved PFS. But it still, of course, remains early, and it remains to be seen if immunotherapy can indeed raise the tail of the survival curve. Finally, I'd like to end by mentioning the new kids currently in the clinic, PKMYT1 and Pol theta inhibitors. You've already heard from Dr. DeRoche about targeting PKMYT1. PKMYT1 is a cytoplasmic membrane-associated serine threonine protein kinase that selectively phosphorylates CDK1 and doesn't actually have any other known substrates, and it negatively regulates the G to M transition of the cell cycle by inactivating CDK1. And Dr. DeRoche, through a genome-wide CRISPR screen, showed that PKMYT1 was the highest scoring druggable hit in a CCNE1 overexpressed synthetic lethal screen, and his group demonstrated that PKMYT1 was synthetic lethal with CCNE1 amplified and FBXW7 loss of function mutations. And this highly selective PKMYT1 inhibitor, RP6306 from Repair, is currently in phase 1 clinical trials. And while CCNE1 amplification is found in different tumor types, the percentage of cancers that actually harbor such amplification is variable. For example, in breast cancer, only 5% of all cases show amplification of CCNE1. However, the cyclin E protein is actually deregulated in about 50% of all breast cancers. Specifically, full-length cyclin E is cleaved at the N-terminus by neutrophil elastase to generate a series of low molecular weight forms of cyclin E that can accumulate in the cytoplasm, as depicted here on the right-hand side by IHC, with a cyclin E antibody that can detect both nuclear and cytoplasmic cyclin E. Importantly, the presence of cytoplasmic cyclin E, which is the low molecular weight forms of cyclin E protein, is indicative of poor prognosis in breast cancer regardless of subtype. And I show here the Kaplan-Meier plots of hormone-positive, HER2-positive, or TNBC tumors caused by the presence or absence of low molecular weight cyclin E. Given these important data, Kanon Kiyomasi and her lab at MD Anderson set out to examine if RP6306 was effective against low molecular weight cyclin E expressing mouse models. To this end, they developed a genetically engineered mouse model where the low molecular weight cyclin E is expressed predominantly in the mammary gland. They then treated two sets of orthotopically transplanted mice with either vehicle or RP6306 BID for 20 days, as shown here. And their results revealed that monotherapy with 6306 was very effective in significantly reducing tumor volume. They then examined the expression of both gamma-H2AX and cyclin E in a vehicle and RP6306 treated mice, and showed that while the tumor retained their cyclin E expression, gamma-H2AX, which is a marker of DNA damage, was significantly increased in the RP6306 treated tumors, supporting the targeting of PKMYT1 in breast cancer. Moving on to Pol theta, this is a key enzyme that's required for DNA double-strand break repair through microhomology-mediated end joining, or MMEJ. This is a multifunctional DNA repair enzyme with both polymerase and helicase activity that's linked by a central linker. And the polymerase domain is thought to be required for all functions, but there is less certainty for the role of the helicase ATPase activity. So there is currently a Pol theta inhibitor ART4215 from Artios, which targets the polymerase activity, which recently entered the clinic. In terms of targeting Pol theta and opportunities in the clinic, There's certainly robust preclinical data now that support the use of pol theta inhibitors in BRCA-mutated cancers and also others with homologous recombination deficiency, and also, most importantly, to target PARP inhibitor resistance. And these will be explored in the ongoing phase 1 study of ART-4215. Promisingly, there are also other pol theta inhibitors in preclinical development from Repair and also from IDEA. So to end, here are my take-home points. In terms of targeting the DDR beyond PARP inhibitors, I think we really now have a massive opportunity to build on the success of PARP inhibitor monotherapy in breast cancer. There are now multiple DDR agents beyond PARP inhibitors for synthetic lethal strategies. There are different patient populations with different molecular subtypes that can benefit. These agents all have different pharmacological profiles and are at different stages of development. There's certainly different combination strategies as well, but patient selection remains critical using predictive biomarkers of response, whether we're targeting these drugs and developing these drugs as monotherapy in molecularly selected cancers or using multiple rational combination strategies. I'd like to acknowledge all patients and their caregivers, also my colleagues from MD Anderson, especially all of my DDR collaborators, my research team, the various grant bodies that have funded our work on DDR, and also multiple industry partners. Thank you very much for your kind attention.